So, what is gene cloning? In general, the word cloning means the creation of a perfect replica. However, in biology, in biotechnology, the recreation of this whole organism is referred to as a reproductive cloning. This one, we clone the whole organisms, okay? But long before attempts were made to clone this entire organism, researchers actually learned how to reproduce desired region, desired fragment, of the genome, a process that we call as molecular cloning or gene cloning. So molecular cloning is the most common type of genetic engineering in biotechnology. Well, we actually clone foreign genes into host plasmid to create a recombinant plasmid. Oops. Why is it so sensitive, this pen? Okay. Recombinant DNA. Alright. So cloning small fragments of the genome allows the manipulation and study of these specific genes and their protein products or non-coding regions in isolation, secara asing, luar daripada the organism punya badan. So in DNA cloning, researchers make many copies of DNA fragment of interest, such as which is the gene. In many cases, it involves inserting a target gene, this is a target gene, into the circular DNA molecule. So plasmid is the circular DNA molecule. Okay, so if you can recall the last image that I've shown you before. So this is another explanation of that image. The insertion is done by using enzymes that cut and paste the DNA. So how do we actually cut this gene out from its original organism? We use enzymes. First, we have to isolate the whole DNA, and then we use a specific enzyme or restriction enzyme or endonucleases to cut. You learn about enzymes in DNA replication. So we use enzymes to cut this out from the original organism, and we ligate them back, using ligase, if you still can remember your DNA replication video, to combine this new gene into the plasmid. Okay, so now you get a new recombinant plasmid or DNA. And then this plasmid, when you transform it back, you transform ke dalam bacteria cell atau host cells. Kita panggil dia selalu host cells. Host cell is usually bacteria. Bacteria kita selalu guna is E. coli. Selain bacteria, we also use yeast. But it depends on the kind of research that people are doing. Okay, so when you replicate this DNA into the host, you can, and then when it this host divide, dia akan ada banyak copies lah. So selain PCR, when you use molecular cloning, you can also amplify the gene of interest. Okay, so in some cases, this plasmid actually codes for proteins yang the bacteria can actually express. Contoh, in the production of insulin using bacteria. Let's say this is the gene for insulin, right? So you insert it into the bacterial plasmid, and then the bacterial plasmid is transformed. So now it has insulin gene inside of its plasmid. And then when the bacteria actually undergo translation process, hope you still remember what translation process is. It meaning you want to convert this gene and make it into a protein, insulin protein, right? When it transforms into insulin protein, dia akan expresskan by this bacteria. So now the bacteria actually produce insulin protein. Kita boleh asingkan, purify this out from the culture, and letak dia dalam vial and bagi dekat diabetic patients. Okay, in a simpler explanation lah, right? Okay. Now, reproductive cloning is a method. Let's go back to reproductive cloning. Like I said, it's like cloning the whole organism to make a clone, to make an identical copy of that entire multicellular organism. So basically, molecular cloning and reproductive cloning, molecular is just a fragment of the genes that is being cloned. Reproductive cloning is the whole entire multicellular organism. Most multicellular organisms undergo reproduction by sexual means, which involve the contribution of DNA from two parents. Half of the DNA, half of the allele from mom, half of the allele from dad, and then when it fertilizes, you get a whole complete set of chromosome balik. Okay. So bila kita when human actually undergo sexual reproduction this way, it's fairly impossible to generate an identical copy or parent or clone of either parent. Maksudnya, look at the offspring, look at yourself. You don't look 100% like your dad or 100% like your mom. You are somehow have these certain traits from your mom. The dominant traits will be expressed, the recessive trait will not be expressed unless you have both alleles. So that's why they are research on reproductive cloning. Okay, to reproductively clone mammals in the lab. The first cloned agricultural animal was Dolly, a sheep, who was born in 1996. The success rate of reproductive cloning at that time is very low. Dolly actually lived for 6 years and it died of a lung tumor. Oops. Okay, so kita boleh cakap dekat sini, clone from reproductive cloning is not as perfect as a human. Okay. But we do have several other species of animal that has been successfully cloned, such as horses, goats, for example. Alright, that's all. Not yet. So let's look into how gene cloning is actually done. First, you have to have a plasmid. Now, the plasmid acts as a vector. A vector is something that can carry this foreign DNA. Now, if you look into this one, dia ada cakap kat sini, this is restriction site. Restriction site is where the restriction enzyme will cut. So it will cut here and here, and then dia akan putus lah like that. Now, when it putus like this, then only the foreign DNA can attach here. How does it attach? Dia guna ligase. Remember in the previous video I said about selecting the transformed bacteria that actually has the recombinant plasmid. So this plasmid actually tunjukkan macam mana kita boleh select. So dia guna ampicillin resistant gene. Ampicillin is a type of antibiotic. Okay, so bacteria that have this ampicillin gene, they can grow on media that contains ampicillin. So bacteria cells yang tak take up this new DNA, dia akan mati sebab ampicillin is antibiotic. Antibiotic can kill bacteria. Okay, so only those with this new plasmid will survive. The agar that contains ampicillin ni kita kata selection media, selective media, selection media, yada sama saja. Fungsi dia ialah untuk filter out cells, bacteria cells that has been transformed with the new plasmid, right? So basically it's actually pretty simple in theory. It's just that when you are in the practical setting, you can consider a lot of things. Some bacteria, maybe all of these host cells won't take up the new DNA. maybe you will have contamination problem. There's just a lot of things you have to consider when you do gene cloning in the lab. Okay. Again, when you transform this into new plasmid kan, into the bacteria host, kita panggil proses ini transformation. So there are a lot of ways to transform bacteria. We can use viral transduction, but the common technique is to use calcium heat shock transformation. Okay, more on that when you are in the third semester taking cell and molecular biology. That's all.